Vacuoles are membrane-bound organelles found in various eukaryotic cells, including both animal and plant cells. While they share some general functions like storage and waste management, their structure and primary roles differ significantly between animal and plant cells. Structure of vacuoles. Mature plant cells typically have a single, large central vacuole that can occupy up to 80 to 90% of the cell's volume. Immature or actively dividing plant cells may have smaller, multiple vacuoles that eventually fuse to form the central one. Animal cells generally have multiple, small, and temporary vacuoles. They are typically much smaller than plant vacuoles and do not occupy a significant portion of the cell's volume. Some animal cells may not have vacuoles at all. The membrane surrounding the plant vacuole is called the tonoplast. This membrane plays a crucial role in regulating the movement of ions and other substances into and out of the vacuole, and in maintaining the vacuole's internal environment. While in animal cells similar to other organelles, their membrane is a phospholipid bilayer with embedded proteins. The fluid inside the plant vacuole is called cell sap. It's a watery solution containing various substances such as water, ions, nutrients, sugars, amino acids, proteins, and lipids, metabolic wastes, pigments like anthocyanins that color flowers, and sometimes toxic compounds that deter herbivores. Whereas the contents of animal vacuoles vary depending on their specific function. They can contain fluid, food particles in the case of food vacuoles, or waste products. Functions of vacuoles. In plant cells, turgor pressure and structural support is arguably the most critical function in plant cells. The central vacuole absorbs water, exerting turgor pressure against the cell wall. This pressure provides rigidity and structural support to the plant, allowing it to maintain its upright position and preventing wilting. When the vacuole loses water, the turgor pressure drops, leading to wilting. Storage. Plant vacuoles serve as a significant storage site for water, nutrients, ions, pigments, and metabolic byproducts. In seeds, specialized vacuoles, also called protein bodies, store proteins essential for germination. pH regulation. The tonoplast actively pumps protons into the vacuole, maintaining an acidic environment within the vacuole, which is crucial for the activity of hydrolytic enzymes and for buffering cytoplasmic pH. Waste disposal. They store waste products that could be harmful to the cytoplasm, effectively sequestering them. This is particularly important as plants lack a sophisticated excretory system like animals. Digestion or hydrolysis. Plant vacuoles can contain hydrolytic enzymes, similar to lysosomes in animal cells. These enzymes break down macromolecules, cellular debris, and even entire organelles, which is known as autophagy. pH regulation. The tonoplast actively pumps protons into the vacuole, maintaining an acidic environment within the vacuole, which is crucial for the activity of hydrolytic enzymes and for buffering cytoplasmic pH. Growth. By increasing in size through water absorption, vacuoles allow plant cells to expand rapidly and grow larger without needing to synthesize a large amount of new cytoplasm. Protection. They can store toxic compounds that deter herbivores or pathogens. Pigmentation. Vacuoles can store pigments like anthocyanins, which give color to flowers and fruits, aiding in pollination and seed dispersal. Functions of cell in animal cells. Storage. Animal vacuoles can temporarily store water, ions, nutrients, and even waste products. For instance, fat cells may have large vacuoles for lipid storage. Waste removal and detoxification. Similar to plants, animal vacuoles can help in sequestering and removing waste products or harmful toxins from the cell. Transport. Endocytosis and exocytosis. Vacuoles, or vesicles, which are essentially small vacuoles, are involved in endocytosis, taking substances into the cell, e.g., food vacuoles formed during phagocytosis, and exocytosis, releasing substances out of the cell. Osmoregulation. In certain single-celled organisms like Paramecium, contractile vacuoles actively pump out excess water to prevent the cell from bursting, a process called osmoregulation. While less prominent, some animal cells also utilize vacuoles for water balance. Intracellular digestion. Food vacuoles, formed during endocytosis, fuse with lysosomes, allowing for the digestion of ingested food particles. In summary, while both animal and plant cells possess vacuoles, plant cells rely heavily on a large, central vacuole for maintaining turgor pressure, growth, and long-term storage, whereas animal cell vacuoles are typically smaller, more numerous, and play more transient roles in storage, transport, and waste management, often complementing the functions of lysosomes.